What is voltammetry? It is an analytical method measuring the current in an electrochemical cell as a function of applied potential of an analyte. There are different techniques in executing voltammetry, such as cyclic voltammetry, polarography, square wave, and differential pulse voltammetry. One example that will be discussed for today is differential pulse voltammetry. So, what do we mean by differential pulse voltammetry? It is an enhanced discrimination of Faradaic currents, electron transferred to and from an electrode, where the potential perturbation, which consists of small pulses, is superimposed upon a staircase waveform. This means that small amplitudes are applied and superimposed at the base potential, which slows down at a gradual rate. It is used for both quantitative chemical analysis and to study the mechanism, kinetics, and thermodynamics of chemical reactions. Current is measured at 2 points for each pulse, before the application of the pulse and at the end of the pulse. It is measured for short intervals after the current is dropped. When the first sample is added, the potential will be read and will be evidently increased. When the first sample stops, other samples will be added and the current consequently decreases. It can also provide improved selectivity for observing different redox processes compared with CV and LSV. This is true because DPV is very sensitive, often allowing direct analysis at the parts per billion level, so one could really observe the improved selectivity to observe different redox processes. It is sensitive because of relatively short pulse time and its differential nature. Often, it is used with a dropping mercury electrode, or DME, or with a static mercury drop electrode, or SMDE. It can also be performed on a stationary solid electrode or on a rotated electrode. Script in the pulse voltammetry software provides for mercury drop generation, solution deaeration, and experiment sequencing suitable for the most common applications for differential pulse voltammetry. We can take a look at where this can be applied. For example, it is very important in examining the pH dependence of redox potential for an electron and proton transfer in tryptophan and tyrosine. The pH dependence is used to calculate the delta G values for different reaction pathways and thus determine that the mechanism can be one-step or two-step depending on several factors. It is also important in examining quantized double-layer charging of hexane thiolate-coated monolayer protected aurum clusters. It provides necessary resolving power by suppressing background currents to separate out all 13 peaks related to aurum clusters' core charging. This helps to make the tough peaks to become visible. This highlights the power of DPV. Moreover, it is important in the determination of DAP zone, also known as 4,4'-diaminodiphenylsulfone, or DDS, in drug substance and product at carbon-based electrode and a glassy carbon electrode. Difference in current is being measured in this and clearly exhibits differential pulse voltammetry. It is also important in the determination of ascorbic acid, pyridoxine, and folic acid in a multivitamin preparation. The individual vitamins all gave well-defined peaks in the anodic region with a linear response of peak current to concentration. The DPV method was found to be generally applicable to the determination of the vitamins in several multivitamin preparations, or in simplified form, to the determination of the individual vitamin preparations. Lastly, it is also important for the quantization of phenols.